Hey folks, and welcome to another edition of Zog Science. Today we're going to be continuing our discussion about evolution, and we'll be getting into the specifics dealing with speciation, or the creation of new species. So what is a species? If we're going to be creating new ones, we probably should have some sort of definition. So a biological definition of a species would be a group of organisms that can interbreed and produce fertile, that's very important, offspring, but they cannot do so with other groups. So basically, you just have a group of organisms going to be able to breed and produce fertile offspring. Fertile means that those offspring can then produce more offspring that can then produce more offspring, so on and so forth. Another important part of this is that there's going to be an absence of gene flow. So there's not going to be any alleles being passed back and forth between this group of organisms and another group of organisms. There are some problems with this definition of a species. First of all, it doesn't apply to asexually reproducing organisms because those organisms, they don't, interbreed at all. So this is really kind of an inadequate way to be able to define different species of, say, bacteria. It also does a poor job of describing hybrid offspring such as mules. Would mules be considered a separate species? They can't necessarily produce fertile offspring. So that's kind of an interesting question. There are some other definitions of a species. First one being morphological. This is basically just based off of body shape, structural features. It is the most common definition of a species, and that is what was used for a really long time. Can be pretty subjective, though. You can have organisms that look very similar, so you might say that morphologically they're the same species, but it turns out that they don't interbreed, so are they actually the same species? We can also have an ecological definition. Here we're going to be taking the niche of the species, and we're going to be using that as kind of the defining characteristic. So the niche of a species is this ecological role, what it eats, what it doesn't eat, what ecosystem services it provides. It can count species as different even if there is some gene flow. So if maybe some groups of a species, perform different ecological functions, maybe they'd be considered to be a different species even if there is interbreeding going on. And then finally, we've got phylogenetic definition, which is the smallest group of organisms that share a common ancestor. So basically, you've got one group that they would have evolved from one specific common ancestor. We would say that's a species. Now, one of the problems with this is that it's kind of subjective where the cutoff line is, right? Technically all life has one common ancestor. So where do you decide where that actual common ancestor is? But these definitions are useful in different contexts and in different purposes because they all have limitations. So why, in terms of biological definition of species, why do we care about reproductive isolation? Well, it allows evolution to occur on the populations separately from one another, which means that your allele frequencies are going to change independently. If you have gene flow, then the gene pools are going to be sharing the same alleles back and forth, which means that whatever your allele frequency is in one population, it's going to be pretty similar to the other population. If there's no reproductive, or if there is reproductive isolation and there's no gene flow, then those alleles are never going to cross from one group to the other, and so therefore you will get evolution occurring on each of them independently. And you can see right here that we've got two different species of birds, and these are different species. You can see some slight differences in their shape, or not necessarily shape, but their coloration. So there are a couple of main types of speciation. We've got allopatric, which means other country, and that's basically where your groups are separated into different geographic areas, and then they undergo evolution differently. They undergo evolution differently, I guess, just because they're in different areas. And then we've got sympatric, which is same country speciation. Here, the species will evolve in the same geographic area. We'll talk about a couple examples of how that could occur here in just a moment. So let's kind of talk about how this isolation could occur. So we've got a group of organisms. They're right here, and they're all, there's gene flow going back and forth. Individuals are sharing their genes. And then we've got a group of organisms that get separated from the rest of them. So it no longer can have gene flow occurring. Therefore, evolution is occurring on it separately. It diverges, and then maybe eventually we have some gene flow that goes back and forth, and we get these hybrids. Now, there are kind of three different ways that this could go. Either our two organisms continue to diverge, and therefore, this is known as reinforcement. So you're reinforcing the isolation. So you get two new species and maybe some sort of third hybrid species. We can have fusion, which is where the gene flow occurs and they come back together again, and now you're basically left with more variation within the overall population. However, it is all one species. Or you could have stability, where you end up with two isolated populations. They are new species. However, they are continuing to breed and producing hybrids. Good example of this would be horses and donkeys, right? They have diverged into separate species. However, they still produce hybrid offspring in mule. Now, in terms of how we can have barriers to gene flow, there are different types. We've got prezygotic barriers, which is before fertilization occurs, and we've got postzygotic barriers, which occur after fertilization occurs. We're going to go through each of these, so I'm not going to read them off for you. The first one is habitat isolation. So basically, species that occupy different habitats within the same area. So this would be an example of sympatric speciation. So we've got these species that are in the same area. However, an example would be the garter snakes. One of them lives in the water, one lives in the land, so they don't come into contact with each other very often. Therefore, they have gone through evolution separately. And most of these are examples of what could be sympatric speciation. Could be allopatric, but most of these are going to be sympatric, or will be exemplified, I guess, as sympatric speciation. The next one is temporal isolation. So these are species that breed during different times of the day, seasons, or years. For example, eastern skunks mate in the winter, while western skunks mate in the summer. So if one is mating at one time, the other is mating at another time, you're never going to mix the gametes, right? Which means that you're never going to have gene flow occurring between the two different species. Next is behavioral isolation. So these are where you have different courtship rituals, along with other behaviors such as maybe songs or other sounds or maybe colorations that help organisms recognize mates. So, for example, blue-footed boobies mate with a unique dance, and so they have this kind of fun waddle. I highly recommend that you go on YouTube and look it up. It's pretty funny, but they have a little dance that they do, and that's how they pick their mates. Other species have their own unique songs or dances. This is an example of a bird of paradise, and you can see that it has this very unique coloration. These are feathers that it has kind of puffed up, and it also jumps and kind of flaps its tail on the trunk that it's on. So lots of different species, especially in birds, but in other species as well. You've got frogs with all their different mating calls that they make. So lots of variation with that. Now. We can also have mechanical isolation. So this is where the mating cannot be completed due to morphological differences. So, based on the structure of the organisms and their reproductive parts, they're going to not, functionally be able to mate. So, for example, these are two different species of snail. You notice that that their shells spiral in opposite directions. And because their shells spiral in opposite directions, their general openings do not align, meaning that they cannot exchange gametes. So therefore, reproduction cannot occur. And this is true in a lot of different species. We also have gametic isolation. This is where we have specific biochemical mechanisms that prevent the sperm from one species from fertilizing eggs from another species. This is used a lot in marine organisms, although it can be used in freshwater organisms as well, but usually it's more marine organisms. That's because all these organisms, or not necessarily all, but a lot of them, they just release their sperm and eggs into the water, right? So if they're just releasing their sperm and eggs into the water, they need to make sure that their sperm are mating with their eggs, right? So, are fertilizing their eggs. So there are specific proteins on the surfaces of the sperm that recognize proteins that are on the surfaces of the eggs. They bind, and then that is what allows the gametes to fuse together. All right, so those are all the prezygotic barriers. Now we're going to be getting into the postzygotic barriers, and the first one is reduced hybrid vitality. Vitality means like health. So this is where the genes of the parent species reduce the hybrid's ability to develop or survive. So salamander subspecies hybrids do not develop properly. So you've got different salamander subspecies. When they mate, the hybrids do not develop properly, so they generally don't make it to past the embryo stage. And if they are born, they are frail, and they're very unlikely to survive into adulthood. Next, we have reduced hybrid fertility. And so this is where hybrid lives full lives, so they're totally vital. However, they are sterile and they cannot produce vital offspring. So a great example of this is a mule, and this is a lot of times it's due to mismatched numbers of chromosomes. So donkeys and horses have different numbers of chromosomes. However, they can mate and form mules that can live, and they're strong. And mules are used a lot, on farms in order to carry out different tasks because they are very strong. However, the mules themselves are sterile and they cannot produce offspring. And then finally, we've got hybrid breakdown. This is where the first generations of hybrids are fine. They're vital. They do have the ability to reproduce. They're not sterile. However, the second generation is frail and they are unable to reproduce properly. And so this is an example of this would be different strains of rice. They have different recessive alleles. And so what happens is that those recessive alleles build up in the hybrid, and then when the hybrid mates with another hybrid, they get more recessive alleles. And so after that second generation, there are too many of these recessive alleles, and these are negative recessive alleles. They are they just reduce the viability of the hybrid, and so eventually you get to the point where you get too many of these recessive alleles, and the organism cannot grow properly. Now, when we talk about speciation, until recently, most scientists believe that evolution only took place over a very long period of time. We're talking about tens of millions, hundreds of millions of years, and this is what we call this idea of gradualism. However, recent evidence has shown that evolution can occur rapidly on a much shorter time scale, maybe like 1 million years, 2 million years, perhaps even less, and we call this punctuated equilibrium. So when we take a look at gradualism, we're talking about species diverging slowly over a long period of time. So here we have one species of butterfly, and slowly over time, they diverge until they turn into two separate species that have, evolved to take advantage of whatever particular environment they're in. In contrast, we have punctuated equilibrium. So here's where the species would have changed very quickly due to some sort of environmental event or other selective pressure. Maybe there was some sort of extinction, so there was new niches that were opened up, or some sort of natural disaster, something like that. And so there's a lot of selective pressure put on the butterflies, so they change rapidly. But then this environment stabilizes, and there's no longer as many selective pressures on them. Therefore, they pretty much stay the same, maybe changing slightly over a long period of time. That's basically what we believe now, is that we have these periods of punctuated equilibrium spaced out by periods of gradualism as the environment pretty much stays the same for long periods of time. So here's an example of allopatric speciation. We've got these two ground squirrels that live on opposite sides of the Grand Canyon, and they can't get to one another because they're obviously on other sides of this giant hole in the ground. Here would be some examples of some hybrids or some gene flow that's going back and forth. So you've got your typical, I'm not going to be able to pronounce this properly, so I'm just going to say the L plants, and then the typical C plants, and these are different species. But you can see that they are able to have hybrids where they will actually breed and not change the shape of the flowers. However, the colors of the flowers will change. So kind of interesting stuff there. Again, this kind of shows you what's going on in terms of hybrids. So we've got these two different toads that live in Europe, and the yellow-bellied toad range is in the yellow on the map, and then the red, the fire-bellied toad range is kind of over here. And we get these hybrids in the middle where we have they share common traits, and that makes sense because you're going to have some overlap in the middle, but then farther away, you're never going to have those hybrids because they're just not going to be able to get to them. And that's all I've got for you today. Thanks for watching, and I'll see you next time.